This article by Lemieux et al. Reports on the application of whole animal screening when compared to in vitro screening of cultivated tissues that are in a basal metabolic state. Their aim was to determine which compounds tested affected the metabolic pathways of fat metabolism in Caenorhabditis elegans. This would indirectly provide insight on how effective these compounds could be when administered to humans. Methods utilized were compound screening, quantitative imaging, and pharyngeal pumping assay. Compound screening placed nematodes at the larval stage in 384 well plates. One compound was added to each plate at a concentration of 5 micromoles in Nile red dye. After culturing, bright field and red fluorescence images were then taken by automated microscopes, as shown here. Pharyngeal pumping consisted of 25 worms placed into 24 well plates with a control substance, or one, of 3,200 compounds at a concentration of 2.5 to 10 millimoles. After culturing, an electronic counter made a count of the number of pharyngeal contractions in 10 seconds. Compounds tested were compared to orally available drugs based on physical and chemical properties. 32 were found to lower Nile red staining and 13 elevated it. Of those that lowered it, 8 demonstrated significant lipid lowering abilities, while 2 increased it. The graph indicates the effectiveness of the different compounds. The report focused on genetics associated with fat storage and metabolic processes in the nematode and how these compounds are affected. Different pathways, though coordinated but separate, were found to affect feeding and fat storage. Compound F17 was examined closer to understand its biochemical capabilities and effects on reducing lipid absorption in cells. As shown here, a specific mutation on the AAK1 gene resulted in F17 being less effective because these worms had no fat storage phenotype of their own. Findings indicate that fat content is not a passive consequence of feeding behavior. Animals with excess feeding rates can have high metabolisms or reduced fat levels in cells. Mechanisms hardwired into the genes or a mutation. This can increase feeding rates to compensate the low lipid levels in their cells. This explains why compound F17 increased feeding rates but lowered fat absorption. Using whole animals instead of cultivated tissue cells allows more accurate measurements of what compounds work in humans. The pharmaceutical industry applies this to develop treatments for metabolic disorders such as obesity, diabetes, and high blood pressure. In this recent article, the Nile red staining method on whole organisms was used to also determine what would modulate fat absorption, with it differing only in the compounds used.